Good morning, everyone. Welcome to the panel section. I'm Jun Xu from Nanjing University of Information Science and Technology, China. Professor Metin Gaiken and I is going to chair this section. So firstly, it's my great honor to introduce the first speaker, Dr. Claude Zenklausen. He is the director of TCGA, National Cancer Institute of United States, and he is also the project officer, NCI Genomic Data Commons, or GDC, National Cancer Institute. The title of his talk is The Cancer Genome Atlas, a Usable Genomic Resource. Let's warm welcome.
Good morning. I see that we have more than 2 people in the audience, which is good. Otherwise it would have been a very lonely moment. So, first of all, I have some disclosures to make. You can tell by my accent that I'm not a native-born English speaker. Which makes my life a dangerous place right now, because we are like, 30 miles from where the bard was born. I'm afraid that the ceiling here will open, and halfway through my speech, a bolt of lightning will make me a little mound of ash. So by the end of this talk, one of the things that may happen to you, because on top of everything not being a native English speaker, my first brush with a place where people spoke English for real was Texas, if we can call that English. Then I moved to the East Coast, which they do speak a more normal version of English, but still. I know, I see some British heads going, it is not really. So, one thing that I can guarantee, if you're an English person or a Scot, is that at the end of this talk, your ears may bleed from hearing me, and this will be the face that the bard will have if he was around, so I'm so sorry. All right. So I'm here to talk to you about TCGA. Probably most of you know TCGA. Oh, I have another disclosure. I'm not a pathologist. Okay, I trained in an animal pathologist lab when I was doing my PhD, but still, I know how to cut a section. Thank you. That's that. I'm a chemist. By training, I'm a chemist. And I ended up in TCGA by a series of fortuitous mistakes. So what we have done in TCGA is shown in this timeline. Basically, 2005, there was a suggestion to NCAB. The federal employees in the States, we talk in acronyms all the time. This is the National Cancer Advisory Board, which is basically our board of directors for the NCI. So there was a report made to the NCAB saying there's an opportunity here to do characterization in large scale. As always, NCAB asked how much. The number was high at the time. And so they said, well, we'll start the way we always start, with a pilot. We'll do 2 tumor types, so they were GBM and ovarian were selected, to see, if you actually could do this. One of the things that people sometimes ask me is why GBMs? It would not be the common first target that you would think of. The reason of why GBMs will be me. I was, at the time, the head of oncogenomics at the neuro-oncology lab branch in NCI, and we were doing a GBM project, mostly with microarrays. We didn't have money for sequencing. We had thousands of samples in our freezer, so we already had the samples. And so we went with GBMs with higher power technologies. And GBM was an interesting project because we started with Sanger sequence targeted sequences, 400 genes, took forever to choose the genes. By the end of GBM, real full-scale exome was a reality, and so we went back to the same samples and redid everything with the exomes, which was a monumental waste of time. But, you know, that's the point of a pilot. You learn what you have to do and how to do it, and so it was successful. The problem was that it took a long, long time to do the first paper. It really was a painful experience. First, because nobody had done in the cancer field a consortium where, you know, what's the authorship model? And then at some point, NCI, we got tired of people squabbling who's first and who's last and who's middle and who's number 322. And we said, enough of it, we're gonna do it in network fashion. Basically, the only author is going to be the network. And that's the way we basically published every single TCGA paper except for the rare tumor ones. And it has worked wonderfully well. All the fears that we had at the beginning that, oh, people are not going to get credit, turned out not to be true. There's people that started as graduate students when TCGA started, and now they are associate professors. And the only things that they have in their publication record is all the TCGA papers. But, you know, when you do something that has some meaning, it doesn't matter if your name is first, second, third, or whatever it is. So in 2008, you probably remember that year as the year of the financial catastrophe. So the States were in risk of a deep recession. The world was in risk of a deep recession, which actually happened, but could have been worse. And so the Congress decided, in their infinite wisdom, to inject some money into the system to make sure that we don't go into a full-blown 1930s depression, okay? And there was a law passed called the American Reinvestment and Recovery Act, ARA, and that was money for projects that were ready to go. Guess what? We were ready to go. And so we got money to push TCGA, and that's when we got authorized to do the expansion to 25 to 35 tumors. We added genomic data analysis centers. Until that moment, the analysis was done kind of ad hoc. We started publishing benchmark papers, and basically we created all the data. Well, we accrued all the samples until 14. We created all the data by 16. And we have been finishing the analysis in the meantime. We did something that I think it was very, very useful for the community, which is the pan-cancer analysis. Instead of having each tumor on its own, we look for themes. We look for things that were common among all 32 of the tumor types. And that was published in April last year. And I think we count that as the end of TCGA. We keep using the samples for new technologies, but the bulk of the program, it's done. So that's why when normally people introduce me as the director of TCGA, I go, well, that used to be. Now, you know, our office has a different name. We have more projects. TCGA was basically the first one that we did in this scale. These are the tumor types that we have. Not all labels are there. Most people don't understand what the labels are. Sometimes people point to one acronym and ask me, what is that? And I have to think, actually, because some of the acronyms are not non-standards. As you can see, they're all in red because we basically published and finished them all. All the ones that are checkerboard are the rare tumor cancers. Some of the rare tumors, like TCGCT, which is testicular germ cell, ended up not being rare after all, because rare means we can get about 100 cases. We got 300 of testicular. We characterized 150. We put the others in the freezer. And now we got money for that, so we're characterizing those. So we have still tumors waiting to get characterized, that they are going to get characterized and added to the collection. There's that lonely blue diffuse large B-cell lymphoma. Some people say that the color is very appropriate because it's sad. We were supposed to do diffuse large B-cell lymphoma, but by the time we started collecting them, the subclasses were very clear, the two main subtypes. And the two main subtypes means that you have very clear therapeutic options. And in fact, nowadays, if you ask any hematological oncologist, they will say, oh, the diffuse large B-cell lymphoma, oh, that's fine, because 95% of them get cured, never to be seen again, okay? There's still a 5% that we don't understand very well what happens. And so what happens is we couldn't get the samples anymore. And so that's why we didn't actually not do diffuse large B-cell lymphoma. We did 80, but that's not a project. Some people say it's like, oh, it's so sad that you couldn't do it. And I go, no, it's great that we couldn't do it. That means people are getting cured. It's like, what is there to be sad? I wish all of these were blue, okay? And that's the point of TCGA, trying to help all of these bars become blue, try to get people to not suffer from these things long. The way we do all our work is through established pipeline, which took a while to figure out how to establish it. When I came to the director's chair, we were still doing things kind of ad hoc. I'm a chemist. I work in chemical industry by the very beginning of my career, and I said that this is not a way to do production. And so I changed the way things go to a stable pipeline. It's not like, oh, where do we send this? If we have this, it goes there. Everything has a point to go. My wife, who is also a chemist and also a scientist and my greatest supporter and my greatest critic, she says that she calls this the sausage-making machine, because tissue enters up there, and data comes out here, and every single thing looks the same, okay? But that's the point. If you are going to compare across tumors, compare across studies, everything has to look the same. And so the process is very simple. We get tissues normally from large research hospitals, sometimes from foundations. It goes to the biospecimen core resource, which extracts the analytes, and sorry, I'm checking myself here. Extract the analyze, gets the data, puts the data into the genomics data commons, and the samples go to the genome characterization centers. We get the data from there. Then we have the genomic analysis network, which are specialists in different facets of the analysis. They get the data, they analyze it. We pull together an analysis working group, which is the clinical knowledge plus the molecular knowledge. We figure out what is in here. We publish the first report. All the data goes to the public. And the first report is that. It's just the first report. The marker paper is showing this is what we did. This is very grossly what we found. Now go at it. Okay? We are not able to do a deep analysis of this amount of data. And so that's why we need the scientific community worldwide to actually take the data and do whatever they want. Sometimes I go, it's like, oh, you're so generous. And I go, no, I'm not generous. This was paid by taxpayer money, so that's not my data, all right? And it was generated with tissues from patients that donated. So I don't grow the tumors, thanks the Lord. So, it's not mine. It's not mine to hold. The problem I'm having is sometimes I deal with larger cooperative clinical groups that they go, my data. And I go, no, it's not your data. It's data that is meant for everybody to use. And so this is the model that we are using in the projects going forward. What are we missing to take genomics AI to the clinic? Well, we need gap closers. And I have this in every single speech that I make, because basic research, the mindset is, I produce the data, I do the analysis, someone else, I don't know who, will do the translation. I don't need to verify if my data is relevant for the patient. And then we have the clinical practitioners that go, yeah, very interesting. I will wait until the FDA, EDA, whoever, approves a drug and test and comes along, and it's not my role to bring this from research to the patient. So we have this gap in the middle that we need to fill. And there's nobody that goes out to university to be a gap closer. So basically, us, basic researchers, need to move a little closer to the clinical side, and the clinical side needs to move a little closer to the research side, and we need to start talking for sure, for real. And so this is basically my call. You pathologists are kind of in the middle there. I'm not saying that you're going to be the gap closers, but maybe is a good place to be. And so here is where we are. We have this big gap in the middle that nobody is feeling, and we're trying to do that feeling. The way we're doing it is by trying to make the data usable. When I got to this position, and they offered me the position, I said, yeah, I'm interested, but I told Harold Barmus, who was the director at the time, I'll take it, but under one condition, that we generate a database that helps people use the data. And he goes, well, but the data is available. And I go, I know, but 3 petabytes of data for download, not many people can do this, okay? We are not doing this for a select few large institutions. We are doing this to pull the brains of everybody that can and is willing to do it. And so the point was, we need to make this available to the biologist that does not have a bioinformatics department attached to their hip. We need to make this available in a way that is easier to analyze. And this is where the genomics data come on. And then Harold said, yes, okay, we'll do that, makes sense. He didn't ask how much was going to cost, which was good, because otherwise he probably would have said no. It's a lot cheaper than we thought originally, because we have a really, really good group that is doing this. But you know, putting a decent database with tools into it that you have to maintain, it's a costly affair. NCI is now deep into this model, and we're going to continue to maintain the GDC and enhance it and all of that. That's a promise that we have gotten. But the whole idea of the GDC is a true tool for the democratization of data. You have all the data that looks the same. It has been processed the same way. It's not like, oh, this was version 3.2 and this is 4.5. We have aligned and called everything with the same colors. And the important thing is we are committed to keep doing that. If the genome build changes, we will realign everything to the genome build. If the tools change, we will recall everything with that tool. People look at me normally like, that's crazy, it's a lot of work. Yes, it's a lot of work, but we put a lot of effort in getting the data, so the data has to be maintained fresh. If we don't do that, the data will age very, 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 very fast. So this is what the GDC looks like. You have different facets. You can analyze the data, access the data, or submit your own data. Access the data, by access we mean download it. Do people still want to download the data? Yes, they do. There's a lot of people that want to get this data. We get hit by robots every single night. And people download petabytes and petabytes. There was an instance when we just had open that we got a big spike of download coming from Lithuania, and we went, huh? It's like, I was talking to him yesterday, it's like, do you know who that was? Because it's like, we don't have any users from Lithuania, but all of a sudden somebody downloads the entire database. And we get this every single night. You know, some people do a delta, an accrual, but there are some people that are downloading the data from scratch every single week. So that's great. Downloading is a way, but what we are trying to tell everybody is like, you don't need to do that, okay? Unless you're a bioinformatician, you're going to use a tool, blah, 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 blah. You don't need to do that. If you're a biologist, you just go and analyze the data in situ. I'm going to go really fast on what you can do. Well, these are some of the programs that are there because TCGA is not the only program in the GDC. One of the things, for example, that we are about to launch out to the public is Project Genie. AACR was going to do their own database, and then they saw what we did, and they go, can we put our data in your place? And we go, yeah, of course. And so we are getting a lot of things. We just released the Multiple Myeloma Research Foundation, 1,000 multiple myelomas. We didn't do it in TCGA because we knew that the MMRF was doing it. And the data had been there, but nobody had been using it because it was in SRA, it was a problem to get. Now all the data is in the GDC, people can actually do the analysis. And so what type of analysis you can do? Well, you start by selecting your cases, okay? So there's a lot of clinical facets, and as you choose the different clinical facets, for example, here we're seeing stomach, you get there the distribution of the most frequently mutated genes. Ooh. That's #1 ask for any biologist when they're studying a disease. They go to the bioinformatician and say, tell me which genes are mutated and what's the frequency. The bioinformaticians hate when you ask them to do that because it's a simple thing to do. And it takes time for them to do it. So they actually love us because now they can go to the GDC. It's like you can pull it out of there. And this is me, okay? And then you have survival plots, but the thing is, you have lollipop plots. When you choose a gene, you will get your Kaplan-Meier for that particular gene. It will divide your cohort into the ones that have the mutations, the ones that don't have the mutations. You can build your cohort whichever way you want. You know, you can maybe say, oh, I want to see breast, brain, and pancreas together. Okay, you're crazy, but that's fine. You know, we will cater to your craziness. And this is done in real time. You don't need to wait. It's amazingly fast. We are wondering when are we going to break the model. We know that we're going to break it at some point, and we're going to have to change it. But for now, it works very well. We are digital pathology, so we do have images of H&Es for every single case because it's a requirement. You don't send the case to us unless you have an FFPE. They are scanned a period 40x. They not necessarily are the prettiest because we didn't make them. We got them from the sites. They either scan them themselves or if they don't have a scanner, we did scan it. But the good news is that even if they are not the prettiest images, it comes with a lot of molecular data attached. So for example, this one is a lung adenocarcinoma that is mutating in EGFR. And this is just a simple section. It's 122 out of 585 lung adenocarcinomas. And so you can choose what you want, and then you can download those images and play with your tools. We are working with Nasir to clean this mess up because we didn't pay attention to the images. I'm a chemist. I care about molecules. You know, what the tissue looks, who cares? And so we had to meet Nasir, and Nasir said, it's like, oh yeah, all nice, but you know, your colors are all over the place, and we can identify what the different cell types are. And Zining, who's sitting down there, and me, we were like, can you do that? And so we're working with them to make this look better and have more value to it. But it's a great resource, and I think it's usable right now because you can check what you want to do without having to, you know, tell a group of 10 people, computer jockeys, do this for me. We did the work. And sometimes people tell me that I'm not afraid to offend the bioinformaticians, and I go, no, they're my friends. I offend people all the time for a living, so it doesn't matter. But they're actually happy with us because those routine tasks that they get asked to do all the time, they don't have to do them anymore. And now they can focus on really complicated tasks that we cannot do at the website. It's not like you're going to discover everything there, but it's the place where you find out what is really important for your research. Okay? So, oops, sorry. That's the portal. You can go play. If you don't like anything, let us know. If you like anything, let us know. We have a really, really, really good help desk. I know I said help desk. They actually help. And they actually help in time because it's in the contract. They have to answer in 24 hours after you shoot them the email. So amazingly, you send something to help desk and you get a response back, and not get a response back when your children have grandchildren already. So this is really useful. Something that I get asked all the time: future programs. I brought this back because Nasir, in the one meeting that we were, says, "Oh, are we getting TCGA 2.0?" It's like, no, we're not getting TCGA 2.0. We already did it. There's a lot of people doing these kind of survey projects through foundations and stuff like that. Why are we going to continue doing that? What we are doing is a lot of new programs that are dealing with clinical grade, real world samples. And so we are taking clinical trials that are finished, we're getting the samples, and we are trying to answer questions that are very obvious. Things like Alchemist is in lung adenocarcinoma. As you probably know, early stage lung, they get resected, they get chemotherapy, they get radiotherapy. 40% of people get cured, the disease is gone. 60% show back again. And here we have pathologists. Show of hands, how many of you can tell by the image of the tissue which one is going to recur? You can't. Show of hands for the molecular people. How many can you tell? We can't, because we never looked. Okay, so that's the type of questions we're going for. In Alchemist, question number one is, we know who recurred fast, we know who recurred slow, we know who never recurred. Why? What are the differences? Because that makes also to treatment, that makes to quality of life. If I don't have to have you every 6 months for a CT scan, and I can tell you, you're done, that increases your quality of life immediately. Yeah? And we're not over-treating, and we're not wasting money in patients that don't need it, and all of those things that we're doing all the time. And so this is what genomics is going to do from now on at the NCI. We're going to try to answer clinical questions that could be answered by biology. So more on that in a couple of years. The big message is cancer genomics is here to stay. It's just a different flavor of it. These are all the people that do great jobs, so I can stand here and look good, although I didn't do anything. And so University of Chicago and Ontario Cancer Institute are our partners in the GDC, and they have done an amazing job. Thank you for your attention, and I don't know if I have like half a minute for a question, but if you have it, if not, I'm staying around. You can grab me out in the coffee break, and I'll be happy to answer any questions you have. Thank you.
Thanks, Zenklausen. Give an excellent talk. Any questions from the audience?
They're going there with a microphone.
Great. Thank you very much for an interesting talk, and thank you for the TCGA resource, which has been fantastic, and it remains fantastic, and we used it for several years on the molecular side, and now more recently on the image side as well. So I have two short questions for you. One is, whether you see any opportunity to actually include further clinical follow-up information, so time on relapses and so on. Mm-hmm. And the second question as well is whether you see any opportunity in the work you initiated now in the image domain to also include some level of annotations. So we downloaded pathology reports as scanned PDFs, which was painful to extract data from. Do you have any plans in that domain to make it easier to use the data?
So for the first one, yes. We are trying to get updates on the clinical data. And the operating world is trying. Problem is some of the people that have contributed samples are commercial firms that are aggregators, so some of them do have more information, some of them don't, so that will be hit and miss. But even the hospitals, there's a very large contributor to TCGA, a research hospital, who will remain nameless to protect the guilty, that has lost the key. They don't know what the TCGA cases are compared to their patient identifier. How can you lose that? I don't know. But so, you know, we go and ask them, and we are paying for the updates. And they say, well, we can't because we don't know. So it will be very much a hit and miss. I think some projects like uveal melanoma, there were 4 groups, there were 4 very involved groups. We are going to have updates on that because I'm pretty sure that these people know, you know, what tumors work what. Some others, God only knows. We are trying to go for the ones that we're going to get more bang for the buck, basically the ones that are more aggressive and kill the patient faster, so we're going to get outcomes with a shorter span of time. Breast, well, we are not even hitting the 10-year, okay? And so it will all depend on the tumor type, but we're making that effort. Oh, the pathology reports. Well, we have to have it in PDF format because they need to be redacted of all information. It's interesting, the first PDFs that we got, they had this black box on top of the PDF, which you can click and remove, okay? And so we told them, no, you know, old-fashioned Second World War redaction. Print it, black Sharpie to it, scan it. And so that's what most of them are. We have plans to extract some information out of it. The problem is we don't really know what information we absolutely need. And that's why I'm announcing here in front of all these digital pathology people, we're going to put together a analysis working group to deal with the pathology issues and also to deal with the image issues that we pay no attention. So we're going to start asking experts, and many of them are sitting here around the room, okay, how would you do this? And so once we get that information, we will try to match it. It's all future projects have the pathology report, have the H&E, have all of that stuff. But we are trying to make it more useful. That's why also all the clinical data has been polished and cleaned, and in the GDC it's easy to search. We want to do the same thing for the pathology side. It's just a little more complicated. It will take a little longer.
These are exciting developments. Let's thank our speaker one more time.